This is a video to help you through the activity on nucleic acids. Nucleic acids are the molecules that we know as DNA and RNA. In the last activity, we discussed the formation of nucleotides from their nitrogenous bases, sugars, and phosphate building blocks. In this activity, we will discuss the condensation of nucleotides to form DNA and RNA. Before we talk about the condensation to form nucleic acids, let's review some of the information from last time. Please pause the video now and try to read and answer the questions on the first page of the activity. First, I'd like to number the carbon atoms in all of the sugars from 1' to 5'. Here they are. This numbering system is similar to the one that we used when we talked about monosaccharides. On the sugars, we designate each of the carbon numbers with a prime. These are nucleotides, and they will condense to form nucleic acids. Each of the phosphate groups will lose an oxygen, and a hydrogen will be removed from the 3' position of another sugar. This will give you an OH group. To make this a complete condensation reaction, we will need one additional hydrogen for each OH group that is removed, and we would get 2 molecules of water. In the condensed structure, I've highlighted the new bonds with pink between the oxygen on the 3' position of a sugar and the phosphorus from phosphate. This is a nucleic acid. Look closely at the structure and determine whether this is a nucleic acid that would be found in RNA or DNA. This one would be found in RNA. I can tell this because each of the sugars has a hydroxide or an OH group on the 2' position. If this were DNA, that OH group would not be there and it would be replaced with a hydrogen in a deoxyribose. This sugar is ribose, and so this is RNA. The new connection here between the 2 sugars is called a phosphodiester because it contains 2 phosphate esters. Because it contains 2 phosphate ester bonds. This one that was formed in the original condensation of phosphate and sugar, and this one that was formed in this condensation reaction as shown. Notice that there are repeating parts here that are the same for each one of the nucleotides. The phosphates are all the same, and the sugars are all the same in this nucleic acid. This is what we call the backbone. It doesn't depend on the actual identity of the nucleotide because every nucleotide in this RNA strand will have the same repeating phosphate sugar. The part that's different about this is each of the bases. The identity of the bases are what we refer to as the sequence of the DNA or RNA. The sequence of bases is the primary structure. Each strand of DNA or RNA will have a 5' end and a 3' end. It's the end of the strand that is not connected. On this side of this nucleic acid, I have a 5' end. This is the 5' carbon of the sugar. Notice I've labeled it over here. It would be labeled the same way here. This is the 5' end of this nucleic acid because this is the end where the strand ends at the 5' carbon. On the whole other side of this strand is the 3' end. This is the 3' carbon of a sugar that's not connected to another nucleic acid. Instead of drawing the entire structure of nucleic acid, often you will write just the sequence of bases. This one would be represented AGC. It is convention to always list the 5' nucleic acid first and the 3' nucleic acid last. Another way to draw the same sequence is like this, indicating the 5' and 3' end. You don't have to indicate which end is 5' or 3' because it's understood that if you just draw the sequence, the 5' end will always be to the left and the 3' end will always be to the right. This is a convention. If you break convention and you want to draw this in the opposite direction, you must indicate the ends. Here's an example. I could not simply write CGA because that would be wrong and it would not represent this particular nucleic acid. I could, however, write CGA if I indicate that this is the 3' end and this is the 5' end. Because I'm not following the convention that I'm drawing the 5' end first, I must indicate the 3' and 5' ends of this nucleic acid. These are 3 different representations for the sequence of this particular nucleic acid. Now let's see if you can use this information to answer number 4 and 5 correctly. If you haven't answered them yet, pause the video now and answer them. Okay, let's see if you're correct about number 4 and 5. The sequence is shown here. And in number 4, the only sequence that matches the structure shown is the second one, AGC. CGA is not correct because it indicates the opposite order of nucleic acids. In number 5, there are 3 responses that are correct representations for this structure. AGC is correct, and so AGC with the 5' and 3' ends indicated is also correct. Here the sequence is written in the opposite direction, but it's okay because the 3' and the 5' ends are indicated. On the last sequence, CGA, notice it's the same order of nucleotides as is shown here, but because the 3' end and 5' end are not indicated, you have to assume that C is on the 5' end and A is on the 3' end. So this is not representing the sequence shown. Nucleic acids form double-stranded DNA and RNA. This is a picture from your textbook of the double-stranded DNA. They show the phosphate sugar backbone inside of this pink ribbon on either side. It represents 2 different strands. The 2 different strands stick together through complementary hydrogen bonding between complementary pairs. The complementary pairs are A and T in DNA, A and U in RNA, and C and G in either DNA or RNA. Let's draw the hydrogen bonding between these complementary base pairs. First, I'm going to draw the lone pairs on the atoms, oxygen and nitrogen. Hydrogen bonding occurs between partially positive hydrogens and lone pair electrons on other atoms. I will now draw all of the hydrogen bonding between the complementary base pairs. A and T have 2 hydrogen bonds, and A and U also have 2 hydrogen bonds that hold them together in double-stranded DNA. C and G have a stronger interaction because they have 3 hydrogen bonds between them. Double-stranded DNA is said to be anti-parallel, and this is because one strand goes in the opposite direction of the other. In the picture shown here, notice that the 3' end of one strand is on this side, and the 5' end is on the same side of the opposite strand. Anti-parallel means that one strand is running in the opposite direction of the other. Notice that these 2 strands complement each other and run in opposite directions. This is anti-parallel double-stranded DNA. One of the DNA strands runs in the 5' to 3' direction. Notice that for every A, there is a T on the opposite side. This is a complementary pair. For every C, there is a G on the opposite side. This is again a complementary pair. You can tell that this is a double strand of DNA because T is present. Even though you can't see the structure of the sugar, you know that T only appears in DNA. Let's provide the complementary DNA strands in number 7. Remember, for every A, you draw a T in the complementary strand, and for every C, you draw a G in the complementary strand, and vice versa. If the DNA is written from 5 to 3', then you will draw its complementary strand in the opposite direction, starting from 3' to 5'. This is what it would look like. Here I start from the 3' end of the complementary strand. For every A, I write a T, for every T an A, for every G a C, and for every C a G, until I reach the end of the strand. I then indicate whether the end where I stop is 5' or 3'. Since I started with 3' here, I end with the opposite 5'. It's very important that you indicate the 5' and 3' ends if you draw the strand from 3 to 5'. Pause the video now and try the second example in 7b. Notice that this strand contains U, so this is an RNA strand. If I were to draw the complementary RNA strand, that means for every A, I draw a U. Here's the complementary strand. Make sure you indicated the 3' and 5' ends and that you matched the bases up correctly. Please pause the video now and try to answer number 8. The nucleic acids shown can be designated in 1-letter abbreviations. First, let's identify the 5' and the 3' ends. This is the 5' end because this is the 5' carbon that is not connected to another nucleotide. And this is the 3' end because this is a 3' carbon that's not connected to another nucleotide. I'll then label the bases using a reference chart. The bases are in the order T, T, G, C, A. And so There are several different ways I can represent this molecule using the one-letter abbreviation. By convention, I can simply write this order and assume that the 5' end is on the left and the 3' end is on the right. You can also indicate this explicitly by writing 5' and 3'. Or you could reverse the order, but if you do reverse the order, you must write the 3' and the 5' ends explicitly. Notice that each of the sugars is lacking an oxygen on the 2' position. That means that this is a deoxyribose sugar, and this is DNA. To draw the complementary strand, first I will draw the actual strand and then I will find its match. This is one way that you could answer this question. The complementary strand is the second strand shown. And for the last part of this question, you can simply write the complementary strand to the strand that's given. This is a DNA strand, and you can tell that because there are T's present. Okay, now let's talk about the structure of DNA. Nucleic acids, just like proteins, have different levels of structure. The first level is primary structure. Just like in proteins, primary structure is the sequence of the units. In DNA and RNA, the primary structure is the sequence of the base units. So primary structure is represented in this figure by the sequence TTGCA. Here's a figure from your textbook that represents the different levels of DNA structure. The primary structure is not represented well, but it is the sequence of bases in the DNA. We're going to focus only on DNA right now. DNA has a secondary structure as well. When two complementary strands of DNA come together, they automatically start twisting around each other to form the double helix. This figure shows the double helix in blue. Each ribbon represents a strand of DNA, and the crosses, as if this were a ladder, the rungs of the ladder, represent the DNA base pairs that are matched between each strand. So hydrogen bonding between two complementary antiparallel nucleic acid strands twisting to form a double helix is the secondary structure. Tertiary structure is higher order twisting of DNA to make compact structures that can fit into your cell nucleus. This is represented by the little strand coming around, wrapping around these things called histones, and then supercoiling into packed structures that can be folded into chromosomes. The higher order twisting is wrapping around histones, supercoiling, and assembling into chromosomes. Really, tertiary structure is everything that makes DNA tiny and compact so that it can fit into your cellular nucleus. Thanks for watching this video on nucleic acids like DNA and RNA. If you have any questions about this video, feel free to post them in the comment section. And if this video was helpful for your learning, please let me know by giving me a vote.